Welcome everyone to the semifinal webinar for the 2023 USABO. We're very excited to have so many people tune into the event, and also all of our speakers here who will be sharing their advice with all of you guys. So from this webinar, you'll be able to learn how to prepare for the semifinal exam, which is coming up next week on March 16th. My name is Leanne Tai, and I will be hosting today's webinar. Before we start, I want to say congratulations to all of the 2023 semifinalists. Whether or not you're taking the semifinal exam this year or you're going to take it in a future year, we hope that this webinar will be very helpful to you in preparing for the exam and making the studying more fun. So we are so honored to have Mrs. Frame, the advisor and consultant of USABO, to guide us here today. We're also very happy to have all of our speakers. Thank you to Kevin, Lucy, Michael, Jason, and Derek, who are all USABO alumni here to share their experiences. All right, so let's get started. Just as a quick overview, today we will be starting off with reviewing the exam structure and the point distribution of the semifinal exam. And then we'll move into some recommended studying strategies and studying resources for the semifinal. And this exam consists of 3 parts, where the panelists will be showing past questions from each part and do a walkthrough and a rationale. We'll also go over how the scoring works for the free response section of the exam, and we will end with a Q&A session. Feel free to send in questions in the Q&A box throughout the session whenever you have questions, and a speaker will be answering it there in the Q&A box. So you can always check the answered question section to see if a question you have has been answered already. But before we get started, I would like to introduce Mrs. Frame, who will be going over some of the login information and reminders. Mrs. Frame is a lifetime educator who has worked with the USABO. She's now working as a consultant advisor. She's the one who is making this competition possible and coordinating everything, and she writes us the emails. So this competition really wouldn't be happening without her. Thank you so much, Ms. Frame. And she will now just be talking a little bit about how to access the exam.
I just want to thank Leanne, all right, because she puts a lot of hard work into this, and each of the panelists for their expertise and their time. Really appreciate it. And congratulations to all of you who are semifinalists here, and to your teachers who are on this webinar with us. Remember, the exam is next week, March 16th, 4 p.m. Eastern Time. All right. I wanted to just go over with you about logging into the Art of Problem Solving to take the exam. You have your username and your credentials. All right. What you need to do. Once you've logged into AOPS, you can use this link and go directly to the semifinal exam. Right. Or if that's not intuitive for you, once you're in there, you can go up in the upper right-hand corner and click on resources, which is highlighted there in the corner. Right. So either way, it'll get you to that. And then the next sheet that you'll see is the resource page. And it looks like this. And when you take that drop-down menu over on the right-hand side, you'll see that there's other contests. You want to click in that. And once you've clicked on that, the next screen you'll see will be 3 different things. They're all math, except there's one that says other contests. This is where the USABO is located. You click on that schedule, and the next thing you'll see will be Lucy. No, is the screen that results. All right. And it's the semifinal exam. If you click to the right on that arrow, that will lead you directly to the semifinal exam. So next week, you can have that site up. And then once the time starts at 4, you can enter your password and start the exam. The next thing that I want to make sure that all of you are aware of is that it also leads to Part C, upload practice session, because that is the essay portion, and that has to be scanned and uploaded onto the Art of Problem Solving site. They will have that session activated tomorrow afternoon, I'm told. So make sure that you do that part of it so you know how to do it. It gives directions exactly of what size file you need to have and step by step, right? And then I want to say, looking forward to working with you. Congratulations again.
Thank you, Ms. Frame. That was a great introduction. And at this time, I would like to welcome our speakers. So we will first be hearing from Lucy, a junior at MIT studying biological engineering. She qualified for the national finals in 2018, 19, and 20, and also received a bronze medal. She'll be going over some resources studying strategies and also part A. We'll also be hearing from Michael, who will be showing us sample questions from Part B and some Part C questions. Michael is a sophomore at Harvard University studying statistics, and he was a finalist in 2020 and 2021, and he earned a silver medal at the national finals. And to finish off our question reviews, we will be hearing from Derek, who will be going through some free response questions and how to answer them. Derek is a freshman at MIT majoring in computer science or bioengineering. He was a 2021 and 2022 USABO National Finals gold medalist and also a silver medalist at the 2021 IBO. So let's welcome Lucy, Michael, and Derek.
Thank you, Leanne, for the introduction. I'm just going to quickly share my screen. A quick thumbs up from the panelists that you can see this. Awesome. So thank you again, Leanne, for the really kind introduction. So as she shared, I'm going to be sharing some tips and tricks for the semifinal, give an overview of the structure of the exam, and also go over a Part A question. So overall, the semifinal exam is 120 minutes and it's online, as Ms. Frame had explained earlier. There's no penalty for guessing, so it's to your advantage to answer every question. In terms of the structure, there are 3 parts. Part A consists of multiple choice questions with 1 answer, and there are 60 of these, 1 point each. For Part B, the questions tend to be more complex. They may have several correct answer choices, and you may have to perform extensive calculations or figure analysis. And in this section, there are 60 questions, 2 points per question, and you get a total of 2 points if all the correct responses are selected for the select all that apply questions. Then for Part C, this part's a little different. There are both short answer and essay questions, and also a few multiple true-false questions. And so overall, the structure is that there are 5 true-false questions, 1 point each. The point breakdown for that is that if you have 4 correct responses, you get 1 point, 3 is 0.6, 2 is 0.2, and 1 or 0 is 0. And then there will be 4 short answer questions, and each short answer question will be worth 15 points. So just an overview of some general preparation tips for the exam. I'd like to usually think of this as building your toolkit and also learning to apply it. And so throughout your studying, it's still important to drill important facts and terminology from your open exam studying and continuing through Campbell's, reviewing important schematics and important terms and vocabulary. And in addition to that, it's important to apply these to real situations. So the first way to do this is through practice problems. Past USABO's semifinal exams are excellent. Open exams can also be good review. Past IBO exams are also really excellent, and they're especially useful for the multiple true-false style questions you may encounter. MIT OCW has a lot of great P-sets from real MIT courses that are really great preparation for USABO questions as well. And one thing that personally helped me was also trying to write your own questions and writing your own correct and incorrect answers so you can understand a topic in a more thorough and roundabout way. Another thing that personally helped me a lot is being able to read and analyze scientific papers. I think this is especially helpful for figure analysis in parts B and C. So the way I would do this is to pick really any biology paper. I'd recommend starting in Science, Nature, or Cell. First, I'd read the introduction to understand the background, and then I'd jump directly into figure analysis. So I'd look at each figure and I'd ask, what's the purpose of this figure? What possible techniques did the authors use in generating that data? And then based on my thoughts on that, I'd compare my thoughts to what the authors actually talk about in the results and discussion section of the paper, almost as an answer key, just to see what I missed. But also just keep in mind that in real papers, there may also be errors in analysis. So what the papers say may not always be correct, but this is still a good potential technique to use nonetheless. And once you're more familiar with this process, I'd also recommend timing yourself. It's really easy to get bogged down in the details of a figure, but on the real semifinal exam and at USABO finals and IBO, you're not going to have all the time in the world. So without further ado, I'm going to go into some general tips and tricks. Maybe many of you have already seen last year, one of our past alums, Catherine Wang, had given a webinar on specific topic areas, what textbooks to use, and also what subtopics to study. So in the interest of saving time, I'm going to go fairly quickly through these, and I'll focus on some tips and tricks for structuring your study schedule rather than the specific topics. So I'll start off saying that it's important to know everything in Campbell's, and it's always good to go back and review. In terms of what to emphasize, I look at the schematics and figures and also review important terminology. Then for the biggest section of the exam, animal anatomy and physiology, I'd recommend studying from Banders as the textbook. And one of the major tips I would have is to study anatomy together with physiology rather than as two separate fields, especially since anatomy frequently dictates physiology. So studying them together will help you structure your studying better and also give you a more integrative understanding of both fields. So to this end, I'd emphasize taking a look at histology and also histopathology to connect the two. I'd also focus specifically on perturbations and how the body responds to them. Useful topics to study under this area include pathophysiology as well as clinical case studies, which are really easy to find online in many different sources like NEJM or others. And in addition, don't forget about non-human and non-mammalian anatomy and physiology. I'd recommend whenever you're studying from Banders, just to stop at certain time points and think about what a non-human or non-mammalian analog would be and to understand the details of that as well. For cell biology, I'd recommend structuring your studying into three major sections: biochemistry, molecular cell biology techniques, and cell biology itself. So biochemistry is the foundation of cell biology. I'd recommend looking at Leninger's. I found this one to be the most intuitive and well-explained biochemistry textbook. I'd focus on biomolecules, proteins in particular, as well as enzymes and their functions. It's also good to understand certain reaction mechanisms, especially in metabolism. And if you need a brush up of organic chemistry to understand those, I'd recommend taking a look at the first few chapters of Klein's organic chemistry and also some carbonyl chemistry. For molecular cell biology techniques, I thought Alberts went over this particularly well. Generally, I think of these techniques as falling into either biochemical or genetic techniques. So those are the two umbrellas that I would mostly categorize my studying under. It's also really valuable to actually structure and categorize the techniques in your head, thinking about how certain classes of assays can apply to different situations. So rather than memorizing specific components of an assay, it's good to know generally how certain classes of assays work and apply them flexibly to different situations or different system modifications. Then lastly, for cell biology, I think both Alberts and Lodish are great textbooks to use. I personally use Lodish, but I've heard that Alberts is really good at reviewing this too. I'd focus on cell cycle, cell metabolism, cell communication, and also cell signaling. And for the genetics and evolution section, it goes without saying that practice is absolutely essential, especially more quantitatively heavy practice. I think one trick to understand is that it's important to apply basic genetics and evolution principles, but to unfamiliar situations. So some examples I can think of are applying Hardy-Weinberg for X-linked traits or calculating the coefficient of relatedness in haplodiploid mating systems. And to this end, I found the questions at the end of each chapter in Griffith's Introduction to Genetic Analysis as particularly helpful to honing these skills. And then for plant anatomy and physiology, Raven's is our standard textbook. This section is fairly heavy on memorization, so it's just something you have to work through. But one thing that did help me with structuring my studying for this is to study anatomy and physiology together with the plant taxonomy. So then when you understand the taxonomy and also understand how specific shared or derived traits arose, the anatomy and physiology for different sections or the different classes and families of plants becomes clearer and more intuitive as well. So it helps save time. And then for the last section, ecology, ethology, and biosystematics, I lump them all together because there's not really a very coherent resource for this, but I find that Campbell's and doing practice questions was very helpful for studying this section. For biosystematics and ecology, I'd review the relevant Campbell's chapters. I think Campbell's covers biosystematics pretty well and in good detail. Specifically, I think ecology can often seem quite overwhelming. I divide your studying into population ecology, community ecology, and ecosystems ecology so that things are clearer in your mind. There's also a quantitative focus to ecology these days, so I'd focus on finding some online quantitative heavy ecology research or PSETs to take a look at and try to understand more. I think those will be useful for the semifinal exam and beyond. And then for ethology, I personally thought past USABO exams, both open and semifinals, were sufficient to practice for this section. I also want to just say a brief word on statistics. It's not a specific section of the exam, but it's important to know, especially for genetics questions and also for Part C. So it's important to know some key statistical parameters and distributions. For example, Gaussian, Poisson, and binomial distributions are quite important. And then terms like mean, standard deviation, variance, standard error, z-scores, p-values, percentiles, sensitivity and specificity, and quite a few more are important to understand. And then for hypothesis testing, it's also really important. Some important ones I can think of include the chi-square test and the t-test. There are many different hypothesis tests, and it's important to know which ones to use in certain situations. And so I'd categorize them by what variable type they're dealing with. For example, a categorical or continuous variable, and then also what parameter is being tested. Is it comparing means or standard deviations or variances, for example? And one book that I found particularly helpful is Modern Statistics for Modern Biology, which you can find online on a Stanford website. It is a bit overkill for the USABO semifinal level, but it's a good resource in case you want to refer to something for help in statistics. Okay, so now I'll go into some tips and tricks for Part A. So remember that Part A is primarily memorization focused and quick fact application. So I would really not dwell on any question for more than 2 minutes because thinking more about it isn't going to bring the facts to your head. And even if you're guessing, since there's no penalty, definitely answer all of the questions, especially since there's only one correct answer, so you could easily fill in some bubbles. I'd also come up with your own answer first, unless the question is asked in a way that the answer itself depends on the answer choices. This will help you avoid trap answers, which that's the way the questions are designed. It's also helpful to have some key schematics in your head rather than words. It's easier to have figures memorized. For example, gastrulation in different organisms, you might see some direct images of gastrulation in the answer choices. Also menstrual cycle fluctuations and hormones. These are just key things to have deep in your mind. And then overall, I think it's important to know definitions and identify those definitions in the question stem and be able to tie the definitions together to form an answer. And so I picked a practice problem from the 2020 semifinal exam that highlights this. This is question 11, and I'll just quickly read over it. So a patient feeling regularly lightheaded and dizzy for several weeks sees a cardiac specialist who discovers that the symptoms arise from dysfunction in a region of the heart that electrically connects ventricular and atrial muscle tissue. This region is so damaged that ventricles electrically respond to atria only around half the time in the patient's heart. And then they have this normal EKG, and they ask, using the information given and ECG above from a healthy person, which of the following ECGs most likely belongs to the patient? And so in this question, I would have identified about 2 definitions. So the first one is this electrical connection between the ventricular and atrial muscle tissue. So that makes me think of the SA node or the bundle of His as an area that could potentially be affected here. And then the second definition I see is that the ventricles electrically respond to atria only around half the time. And so this is a subclass of second-degree heart block. So I know in the answer choices, I'm going to look for something like that. So based on these definitions, I'm going to come up with an answer I expect. So if I expect second-degree heart block, I'm expecting that QRS waves don't always follow the P waves. And in this case, it's probably going to be in a 1-to-2 ratio since the question specifically states responsiveness half of the time. Then I think about some trap answers I anticipate. I'd only do this if you actually have time. So first, I'd anticipate rhythm issues rather than conduction issues. For example, tachycardia and bradycardia. And then I'd also expect confusion with other types of heart block. For example, first-degree heart block, which has a long interval between the P and QRS complexes, and also complete heart block, which has completely independent appearance of P and QRS waves. So here are the answer choices. So already, based on what I just explained, I can eliminate A and B. A is tachycardia and B is bradycardia. And what I actually notice is that the P and QRS complexes in both of these cases are coherent and unified. The only difference compared to the normal EKG is the rate of the stimulation. And so I can eliminate those two answers confidently. I'm looking at C, and I think it is following some of the patterns I expect, so I'll come back to it. But for D, I can identify that this is first-degree heart block, one of the trap answers I anticipated, since actually the P and QRS complexes are all quite coherent. It's just that in the latter two heartbeats, you can see that the P wave, the QRS wave follows the P wave only after a very long gap. And so that's characteristic of first-degree heart block, not second-degree heart block. Then for answers like E, none of these ECGs could belong to the patient. I generally would not recommend picking this unless you really can pinpoint why everything else is wrong. And so I'd avoid that unless you're really sure. So then coming back to C, this is following what I expect. I see these P waves that are circled that aren't followed by a QRS wave, and it happens every other beat. And so this is exactly the type of second-degree heart block that I'd expect based on the question stem. And so there you go, you have your right answer, and that should take less than 2 minutes. So that's one question. Definitely other questions are different. Happy to answer anything in the Q&A, and I'll pass things on to Michael for part B.
All right, thank you so much, Lucy. I'm just gonna pull up my screen and talk about parts B and C. All right, so Lucy gave some great overview of how you might want to prepare for the exam in general, great textbooks, resources, recommendations, as well as problem-solving strategies for part A, but a lot of them also apply to how you want to confront questions in part B and part C. So just summarizing some of that overview of what might be the same or different, one of the things that you might notice is that part A is primarily multiple choice with one answer. It's very much concept checking type questions, right? Do you know about different types of heart blocks, things that you might want to read about in like Campbell's or Leininger's, etc. While part B might have more complicated complex sequenced multiple choice questions. So sometimes you'll have a large diagram and 3 types of questions that follow from it. You might have just more involved questions that involve not just do you recall fact X and fact Y, and more so like how can you apply this to solve the problem that is in front of you. And then finally, part C sometimes has multiple true false questions as well. And these types of questions are often also more complex than part A and part B, but maybe less so of like a kind of what I call like a narrative question than what you normally see in part B. So I think just on top of the wonderful things Lucy has already recommended doing on the exam, the things I want to emphasize are that, first of all, this exam is very much a time crunch. There are so many questions in so few minutes that you might not be able to come back and review questions. I know when I was taking the exam, some of the things I used to think about were like, can I get through the entire exam in time? Can I say, I'm not sure about this question, mark it and come back to it later? What I often found, and maybe you're faster at me than taking exams, is that there's very little time to come back and review questions. You will probably just be doing new questions the entire time. And so it's important to be able to decide, I think I've spent enough time on this question. I think if I spend the marginal minute if I spend 1 more minute here rather than starting on a new question, I might not actually earn many more points. And so this is a very good distinction to make, that you might not be able to come back and improve on a question. Certainly you might have to do this for part C, for example, but it might not be as worth it for like part A with 1 point questions, or part B where each question is worth individually not as much as well. Other things that might be important are that Part B looks a lot like Part A. You have multiple choice questions, but Part B is usually worth double the amount of Part A. And so an interesting idea, and I'm not saying that this is necessarily the best recommendation, is to think about Parts B and C first, maybe. And Derek will talk more about the Part C short answer question, but one thing that I found helpful was just taking a look at what the Part C short answer questions were in the past and moving back. I think this is also helpful for Part B, multiple choice questions as well, and Part C, looking at the kind of high yield point rich questions first. And then if you have time doing Part A, if you run out of time, then you're losing less potential points than you might be at Part B and Part C. Now, notice that this is still a trade-off, right? Maybe Part A is easier for you to do. The questions are usually less involved. If you have a very good understanding of the facts, maybe you can speed through Part A. And Part B and Part C might be a slog in the sense that you might get stuck on a question, you might get stuck on a complicated calculation, but otherwise, all things like considered, I think too often people take exams front to back, and there's not always a requirement for this. And so I just urge all of you to be mindful of the fact Part B and Part C, in fact, have more points. And finally, since Lucy had talked about how to evaluate a question by thinking about what you know, looking at what's been presented to you, kind of applying your knowledge, I wanted to focus my part more on how do you approach questions where you have very little knowledge. So the best case scenario with answering questions is that you've seen this question or you know exactly what it's testing about. And because you know exactly what it's testing about, you can just answer the questions. They give you this graph, it's beautiful, you already know how to read it. They give you kind of a story about an experiment, and you've seen a very similar experiment before and you can answer it. But there will certainly be questions as well where you're not sure how to proceed and you want to be able to kind of approximate out the solution or make a best guess effort without spending too much time. So that's what I will try to focus on. So working towards this approximate solution, first of all, you do still have to know things. I'm not encouraging this strategy as you don't need to study any biology at all. You walk in, you're the best at approximating every solution. This would be a wonderful thing, but what you'll probably find out is you need a baseline level of biology to just read what the question is asking and to get important facts that you can work with. And we'll see this more as we go through these slides. And then I think another thing with multiple choice is thinking about what is likely or unlikely to be true. You might have heard this in things like SAT preparation, right? If you see the words like all or none, these very absolute terms, just one counterexample or exception to statements like, all, I suppose like all like animal like erythrocytes have no nucleus. Like the word all is a big red flag, because if you can just find like one very obscure counterexample that is not the case, then it's probably not true. And so you want to be avoiding absolutes, but this is just a larger example of, thinking about what is likely to be true and what is unlikely to be true. And this also ties into what I have found to be a very helpful kind of strategy this entire time, which is what are the question writers trying to see if you understand? I think you should think of the exam from the exam writer's perspective. You should think, they're testing me about erythrocytes. Like, why are they testing about erythrocytes? What are some important facts about erythrocytes I know that they might be trying to get at? These are probably useful ideas. What are things I remember from Campbell's? What are things I remember from like Vander's? These are important ideas that you might want to use because maybe the question writer was thinking about these concepts when they were writing the questions initially. And again, we'll see this all applied now. I've been talking quite a bit. We'll try to apply these to an actual Part B and Part C question from recent years. So I think this is a very quintessential quantitative Part B question that looks big, but I think is high point potential. Looking at this immediately, kind of with no real preparation, what I see initially is that there's a lot of numbers. I'm not exactly sure, for example, what all these formulas are. Is mortality rate proportional to the starting population? Is it proportional to the population in each age class? What is an age class? How do I calculate any of these numbers? They all look related. And so there's a lot to digest initially. I think a lot of answering these questions, these larger questions with figures or graphs or diagrams, really 80% of the challenge is sitting down and going, I can read this. I am able to read this. It looks like it's a lot of content, but I can digest and get through it. And so looking at this, I see this looks like, there's different age classes, there's number alive, number dying, proportion surviving, mortality rate for country. I'm going to apply Lucy's hint, and I'm going to maybe just look at this table first. The fact that they labeled A, B, C, D, E, F, G, H, I, J, K, L, M, or not I, but I think this implies that I'm going to need to know what all of these letters are. And so maybe I'll take a stab at trying to solve for these first, and then seeing if I can use the information from this chart to kind of solve the rest of the question. So the first thing is I'm not even sure how to start. I see number alive is missing in H class 0. How am I supposed to get that? Not incredibly sure. I look around, there's number dying. I'm not sure if this means like, is number alive plus or minus the number dying? Like, does the number dying die off after this age class, or does it die off before this age class? So, I mean, but just looking and getting an idea of how this works, I'm trying to look for the most complete row on this table to get information on. Here are some observations I make. In age class 0, 100% of the proportion is surviving. So this must be a proportion with respect to the initial population. That's to one of the original questions I asked. Other thing to notice with mortality rate is decreasing over time. It's actually getting extremely small by age class 2, 0.042 mortality rate. So it's very likely that this mortality rate is with respect to the initial population as well. You know, I was thinking maybe this mortality rate, this, like for example 0.0818 is with respect to or this is a bad example. Maybe like 0.042 is with respect to B. But this is very unlikely because a mortality rate of 0.042 is a very small number. And so it's much more likely that this is a mortality rate with respect to A. And this is, again, from the mind of someone who is not sure what a mortality rate is. Like, I have this idea, right? Mortality means death. It's a rate of how many animals are dying, but I wasn't sure what the denominator of this was. And so I'm trying to work out what is the most likely outcome here. So yes, you still need to know what a mortality rate is in general. You can be somewhat confused about what the denominator is, but you can't be like, I don't even know what a mortality rate is. Then you can't apply the concepts here. Fecundity does not seem very useful to me right now. I was thinking maybe fecundity, oh no, maybe like the population is increasing at every step, but that doesn't seem to be the rate either. It seems that it's just dying off at every step. So now my idea of this chart after this minute or so has been, okay, so I'm starting with an initial population of A, and every time in this generation, certain number dies. And as you can see, this becomes the next generation. The reason why I know this is the case is because I see 95, 37, and 58 in age class 3 and 4. And I noticed that 95 minus 37 is 58. That's why I know fecundity, which is labeled 5 on this row, is not relevant to what I'm doing because I don't see an increase. That's also why I know that I can just get number alive of next generation from the number alive minus the number dying of the generation before it. So great. I have pretty much all I need to solve for this graph now. My initial confusion was really just about interpreting what is a mortality rate, what is the proportion surviving. But by looking at the information I had and making estimations like, oh, I think 0.042 is too small of a mortality rate to be with respect to the current generation, it's probably to the initial generation, I'm able to work and complete this chart. And so I start by saying, looking at the mortality rate of 0.818, and I said that this is with respect to the initial population. So I multiply 1 over 0.818 by 982, I get 1200, this is the initial population. And then the remaining B, D, E, and C, we talked about how we can just take number alive and number dying of generation N and use that to calculate N plus 1's number alive. So we're just going to do that and calculate all these numbers. This is just addition and subtraction, so you can do it by hand. I think you have a calculator as well, but this next part you will need a calculator for, right? I have all the number alive and dying now, and now I need to calculate proportion surviving. So what I posited was that the proportion surviving is the number on number alive divided by the initial population. Why was this the case? Because I saw that in age class 0, proportion surviving was 1. This is probably 1200 over 1200. So to calculate F, I'm going to do 95 over 1200. And as you can see, I get 0.079. I can fill out the rest of this. And the mortality rate we said is we're going to be with respect to 1200 as well. So we fill out all that. Great. Okay, the table is solved. I had some confusion in the beginning, but I had pretty confident in my answers now. As I filled these out, it seemed very likely that the numbers I'm putting down are right. And I can just look, there are some trends with the proportion surviving. It seems proportion surviving is going down. Mortality rate is going down. Like this looks like it would make sense. So now we're going to take a look at these statements that are true. A, the species is most likely R-selected. Again, if you don't know what an R-selected population is, like nothing can save you here. You do need to know what an R-selected population is. This is covered in Campbell's. Maybe I have a good memory and I remember this figure that is so beautifully provided by Campbell's. And I remember that in R-selected species, we'll see a very high mortality rate at a younger age, and then this will kind of level off over time. Versus K-selected species, you can always think like humans or elephants. Very high survival in childhood, and then you see much more of mortality coming in later generations. And so looking at the mortality rate, it looks like from the first generation alone, like 81.8% of the population just dies off, right? We had 1,200 people in age class 0, and these aren't people, these are animals. 1,200 animals in age class 0 and 218 animals in age class 1. That seems very R-selected. I guess one of the things that I'm using as a heuristic is like, would I imagine in a human population of 1,200 that 800 of the humans die at age 0? And my answer is probably not. And that's just based off intuition. And so the species is most likely R-selected. I agree with this. Most likely also makes me feel good because it doesn't say the species is definitely R-selected. Most likely gives me leeway to say this is okay. This is a likely answer. B, the initial population was 1,200 individuals. Wonderful. We've already solved that. That's true. Values B and D are 145 and 50 respectively. This is true. I actually feel very good that this matches up because it probably means I'm getting around the right track. These are very specific numbers they're providing me. Values G and J are equal to 0.048 and 0.061 respectively. And if we just look at what we've done before, this is true. And then values H and M are equivalent, and this is true. So again, we kind of just fill out the figure first, and then we looked at the choices and we decided based off of the figure what to do. This is what you can do in a lot of graph and figure and chart things. Try to figure out what the graph or chart is saying first. Maybe you get stuck somewhere. For example, when I was doing this right before the webinar, I actually calculated the number for H incorrectly. I just said that H was 0. I forgot that we were using 8 over 1,200. And so when I got to the choice of E, and I thought this was not equivalent, it made me double check to make sure my numbers were right, essentially, because I had a suspicion that maybe I made a false calculation, proportion surviving. And so sometimes you can use the answer choices as a crutch to correct yourself if you get things like B and D are 145 and 50. If you had 145 and 47, maybe this is the time to self-correct. Maybe you're right. Maybe you do the calculation again and you're right, but maybe you're wrong as well, and this is a good reminder. So that's an idea for how to approach kind of a larger question that's more involved before you can start answering questions. But you should feel good when you see these questions, because if you can just get the figure and the chart working, there's usually a wonderful load of points waiting for you based off your interpretation. A sample part C question that I picked was this one. Central dogma of molecular biology states that DNA encodes the messenger RNA, okay, which in turn is translated into proteins by the ribosome. Cool story. I'm yet to see why the story is interesting or relevant to this specific problem. So let's find out. One interesting difference among different organisms is how many proteins can be encoded by a single mRNA. So I think this is probably relevant to the question. Otherwise, they wouldn't tell me this. Indicate which of the statements are true or false. First of all the prokaryotic mRNAs are monocistronic. The suspicion for me here is the word all. I don't like the word all. Maybe prokaryotes are very interesting organisms. There's probably some crazy prokaryote living in a hot spring somewhere that behaves differently than you expect it to. And for that reason, I'm already very suspicious of this. And I'm likely to mark it false. Hopefully the background knowledge you come into with this is that you have this idea that prokaryotes can have polycistronic mRNA. You have this idea that eukaryotes are generally like monocistronic. And so, okay, so I say false here because of the word all. It makes me suspicious. Most of the eukaryotic mRNAs are polycistronic. This one you probably just want to know that this is not the case. You can maybe think polycistronic and monocistronic. Maybe I'm not sure. I have these ideas. Maybe I can work monocistronic, polycistronic to actually tell you the definitions, but you can use root words as well, like mono and poly, to figure out what they mean. I have these ideas about prokaryotes. They have things like the lac operon. Maybe I studied the lac operon in chemical biology. Looks like it has this one, mRNA. It's huge, and it codes out a bunch of proteins that are related to lactose transport and pretty much anything that has to do with lactose when it's in the environment. So I have this idea that maybe prokaryotes are polycistronic, and then eukaryotes are probably monocistronic then. And this would maybe make sense with the central dogma I was told here. There's one DNA codes the messenger RNA, which in turn is translated into a protein. I'm told that there's a difference actually between them. It says one interesting difference among different organisms. So I should think that eukaryotes, maybe in this context of this question, are different than prokaryotes. C is another interesting application question. The genome of chloroplast in photosynthesis generates mostly polycistronic mRNA transcripts. So as of now, again, my baseline knowledge is that we have eukaryotes and they're like monocistronic generally, and you have prokaryotes and they're like polycistronic generally. And now I have to apply actually from maybe a different area of study I thought about, which is that the chloroplast and the kind of the big gulp theory, there's a much more elegant way of talking about it. And so, okay, I have this idea that chloroplast maybe the leading theory is that it was a prokaryote that was engulfed by eukaryote originally. And so, it would make sense then that this chloroplast, which has prokaryotic origins, behaves more like a prokaryote and generates mostly polycystronic mRNA transcripts. I like that suggestion because the reason why I think this is true is because I applied concepts I know for a fact to this question. I have a solid chain of reasoning. The more I reason through this, the more I believe that my answer is correct, because this seems like the type of interesting kind of logical chain that I would have to use for this question. Finally, internal ribosomal entry sites are used by eukaryotic viruses to generate polycystronic transcripts. Okay, so maybe I don't know what any of these are. I have this idea of how transcripts are being, translated, or how kind of eukaryotes are working. We said eukaryotes maybe mostly monocystronic. Maybe you want a polycystronic transcript. I can totally see why this is the case. Internal ribosomal entry site. I'm not sure what this is. I have this idea of the ribosome. Maybe the ribosome, eukaryotic ribosome, normally starts to look for like the 5' cap on an mRNA. And okay, maybe in these viruses, they don't have a 5' cap. You might want to know this from, like, for example, bioengineering. This actually does show up where you can engineer transcripts such that the ribosome will attach at a different site. But internal ribosome entry sites are used by eukaryotic viruses to generate polycystronic transcripts. I don't see anything internally like wrong with that per se. And here's maybe I don't have the factual basis for how to approach this question. Maybe this is my first time seeing internal ribosome entry site or polycystronic transcript. But like I could see that maybe there is a virus that does this. Maybe there are some, in a sense, evolutionary advantages for viruses that need to do this. And this sounds like a good idea for getting your transcripts. So I'm not sure here, but I'm more leaning towards true. Now, again, before we wrap up, if I knew the facts, if I just knew how to answer every single question here, this would be great. I wouldn't even have to like think about this. I'd be like, yeah, okay, I know this is exactly what the genome of the chloroplast does. But what I'm stressing here is that there is, in fact, a way to try to work out responses, even if you're not entirely sure what you're working on. Yes, you'll need to know baseline knowledge. You should know what an mRNA is. You should know maybe like the big gulp theory. You should know about prokaryotes, but you don't always need to know 100% of the information, and be able to adapt your knowledge on the fly and think about what sounds reasonable and what doesn't is probably what makes a difference in terms of earning lots and lots of partial credit. So yeah, that's the end of my segment of the presentation. Part C is not just true false questions, it's actually mostly essay-based questions, and the wonderful Derek Chen will get to speak more about that.
Okay, hello, yeah, so let me share my screen. Okay, yeah, so yeah, I'll be talking about the free response part of part C and some like strategies and tips about the part in general, as well as going through two different past USABO part C open response problems. So yeah, let's look at one of these problems. So yeah, this one will be sort of a graph reading problem, and the other one will be sort of like more like logic and also like information-based problem. So for this one, basically they give you two graphs of sarcomere length for muscles versus tension, as well as tension versus velocity, and they also have a massive wall of text. And so just in general about free response questions, so even with all questions in general, oftentimes if there is a large amount of text, you should not like automatically be scared by a large amount of text, because they will often give away necessary information in the text. In fact, like sometimes the amount of text is actually correlated, like more text will mean an easier problem because they like give away more like essential information in the text of the problem, rather than assuming that you already know the information. And so, for example, in this problem, some important things that they give away is that, first of all, obviously they describe what the graphs are, but also we'll use things such as it has been determined that the maximum velocity and the force velocity curve depends only on the intrinsic speed. We'll use like other important tidbits from this paragraph in order to answer the question. And then so another tip that I have for mathematical and graph-based free response questions or just questions in general is you should always, for graphs, you should always notice the intercepts. And then for other like mathematical equations and stuff, you should also basically try to test things to the extreme. So for example, some of you might know of the Michaelis-Menten equation. So you might think of like as the substrate concentration goes to infinity, what will be the behavior of this equation? Same thing with these graphs. You want to think one quantity is 0 or one quantity is infinity. How does the graph behave? And what can that tell you about the problem? So for example, in this problem, basically what they do is they have oh yeah, another important thing is that you should try to understand the experimental setup. So yeah, like especially for when they give you like a scientific experiment, you should like try to think about how the experiment is structured and why the scientists made certain choices when designing the experiment. So yeah, in this one, basically, they have two different graphs. One of these graphs is they are holding the muscle fiber at different lengths and not allowing it to shorten. And then in the other graph, they are holding it at a certain tension, and then they are measuring the velocity in the muscle at a certain tension. So going back to our idea of noticing the intercepts of certain graphs, for this one thing we might notice is that if the velocity is 0, that basically corresponds to the fiber not being allowed to shorten. And therefore, when the x-axis of this basically like corresponds in a way to this graph. Because in this graph, we are not allowing the muscle fiber to change in length. And then that is the same thing when the velocity is equal to 0. And then also, this basically, they make us choose a curve. And then, so this is basically sort of an MCQ with justification strategies. But yeah, you should always try to like think about what are possible answers to this question, and then try to rule out things that don't make sense. So with that, this is how we would solve the problem. So they want which curve. So we know that curve A corresponds to a length of a sarcomere that is 2.1 nanometers, or sorry, micrometers. And then now we want to figure out which of the following curves corresponds when the sarcomere length is set to 1.6 micrometers. So generally, they won't give you extraneous information. So here, in this case, 2.1 would correspond to about here. So what that means is one, for the first graph, we know that it is an isometric contraction. So when the sarcomere is held at a constant length and not allowed to shorten at all, the tension in it will be around 0.25-ish. And then on the other hand, what is the force velocity curve observed when the sarcomere is set to 1.6 micrometers? Even if we don't know the exact numbers, we can see that clearly it is less than the original tension. So what that means is that, therefore, the place where the curve, where our desired curve intersects the x-axis on the second graph should be less than the original curve, because we know the original curve has a higher isometric titanic tension. So therefore, this point where it intersects on the original graph, we're searching for a lower one. So in this case, we can basically rule out A, B, and D, because these all intersect at the original point. And right now we're looking for a lower one, so like around 0.15. So now we're just down to just choosing between C and E. And to choose between C and E, we have to use basically information that they give us in the the text. So it says that it has been determined that the maximum velocity depends only on the intrinsic speed. So this keyword here, intrinsic, it means that it's just a property of the myosin itself rather than a property of the sarcomere length. And therefore, we know that the maximum velocity, so maximum velocity corresponds to when there's zero tension in the sarcomere, or you can also just think of it as like the maximum point on this graph. And yeah, the maximum point on this graph should be basically the same for all these different sarcomeres because it is an intrinsic property of the myosin. So therefore, the correct answer is C, because C intersects the original line A at the y-intercept, and it also has a lower x-intercept corresponding to the lower tension when the sarcomere length is shorter. And then finally, we just have to make sure we explain our problem well. So yeah, Michael mentioned a good thing. So like try to think what the test writer is testing you on. But yeah, oftentimes in free response questions, you like, in order to explain your thinking, you should try to include keywords and key ideas that you think the test writer is trying to go for. So yeah, in this case, yeah, you can like drop things such as isometric contraction to let the test writer know that you understand what is happening. So yeah, so for this one, we would just say that using the left graph, we can tell that when the sarcomere is held at a constant length, the tension should be lower for sarcomere length that is lower. But when there is no tension and the velocity is at its maximum, the intrinsic speed, the velocity should only be dependent on the intrinsic speed at which the myosin heads can complete one cycle, which should be the same regardless of the sarcomere length. And so here is another open response question. I think this is from the 2018 USABO semis. And so here again, we're going to try to, this one is more, it's not like graph reading in the same way, but it also has both mathematical and knowledge-based concepts. So for this one, let's start with some observations. So this is a multi-part question. And again, we have explanation parts for the first two. Therefore, we want to try to throw in key terms for the grader. So basically, if they'll see the key terms and know that you know what you're talking about. And also it helps shorten our response. So the main idea of our response does not get lost in a really lengthy explanation. And likewise, as with Michael's problem solving with section B, you don't have to like know everything in the problem. And also, if you can't do a part, you should always move on. You should try your best, but it's not worth it to like spend like a large amount of time just working on one part. And so yeah, for the first two parts of this question, it is heavily knowledge-based. So if the YAC DNA is highly methylated, what will happen to the efficiency of restriction enzymes? Restriction enzymes are affected by methylation and have like lower recognition basically when the DNA is methylated, or they generally can't recognize methylated DNA unless it's a specific type of restriction enzyme. But even if you didn't know that, you could basically think restriction enzymes are very specific to their target sequences. So if there's any changes to the target sequence, such as methylation, that could often result in a decreased efficiency. For part B, describe how restriction enzymes got their name and what is the biological relevance of restriction enzymes. So yeah, for this one, the first part, is just actually both parts are knowledge, but the first part might be a bit more obscure. Restriction enzymes get their name from the strain of the bacteria they were isolated in. But even if you didn't know the first part, you should always like try your best. So in this case, if you happen to know the second part, the function of restriction enzymes, and important to mention immune systems of bacteria in order to sort of chop up viral DNA. But yeah, now we have the logic based part. So yeah, if you weren't able to have the information to complete parts A and B, you should still try your hand at all the parts of the problem. So for this part, it says provide a restriction map for this YAC clone and indicate the distance between successive restriction sites. So if you know that a restriction site is just the distance or like the place where a certain enzyme cuts the DNA. In fact, they mention it gets cleaved by these two. And so you can think like cleaving is like cutting. And then if you didn't know what a restriction map is, you can sort of guess that it's like basically a map that just describes where all the restriction sites are with their respective distances. Likewise, it definitely helps to know that YAC stands for yeast artificial chromosome, which is a type of circular DNA. However, if you didn't know that, there are still, you could still do this problem because if you tried to do it with a linear piece of DNA, it wouldn't actually like work out. You wouldn't be able to do it. And then the next thing you would think probably, I should try it on a circular piece of DNA. But so regarding how to actually do this problem with a circular piece of DNA, strategies just in general are, first of all, we're going to find the total length. That's pretty easy. We just add all of these up. And just like testing, just like general problem solving skills. But then, one of the important ways I would like to do it is I would break this up into parts. So I would want to find the relationship between the HN sites, then the SAC sites, and then the XHO sites, and then try to basically overlay them into pairs. So with these like complex problems, it's generally good to consider like one feature at a time and then try to see if you can combine them into a larger one. And specifically, we're going to try overlaying SAC1 with the other two because it has less fragments. You can see it only cuts it into two versus these three cut it into 3 different fragments. If you're confused, even though it says YAC DNA, it has telomeres, it is actually still a circular type of DNA because it's an artificial type of DNA. But yeah, so basically what we can do is first finding for each individual restriction enzyme, finding the restriction sites is pretty easy because they literally give us the distances between them. So we start out with like basically these 3 circles, and now we're going to try to overlay them so that they match up. So for example, let's start with trying to overlay HI with SAC. And then one thing to try to see is that basically this 0.5 segment probably corresponds with this 0.5 segment. Because like otherwise it would have to be chopped up into even smaller parts. So that probably and we don't see any like smaller segments on this gel. So therefore these two will correspond with each other. Likewise, we have a 2 kilobase pair segment here. It probably corresponds with one of these, And then finally, we just see that the only thing that happened was this 4.51 was basically chopped up into two, a 2.51 and a 2.1. So what we can do is on our hinge thing, there's two possible places we can cut it. We can cut it so that this is 2 and this is 2.5, or we can cut it so that this is 2 and this is 2.5. And then now we just have to figure out a way to get SAC into this, and only one of these should work with the addition of SAC. And in this case, if we put it one of the SAC sites here, you might also notice that this 2 and this 2 can add up to 4, which is one of our lengths for the SAC digest. So therefore, if the other SAC site is there, then it will work out. And we've completed our map for basically these two at one time. And we can do the same thing for these two. You should get this restriction map. And then finally, to combine the whole thing, you want to think of basically we are trying to match the pink lines here, and you can think of like rotating the circle until these pink lines match, in which case the final map should look like this. But yeah, so we were able to solve this problem by starting with smaller pieces and then basically like putting it together into a larger one. And this would be our final restriction map with all the, I have it color coded, but on the exam, you should probably and for this problem, I don't think it will work with linear DNA, because for it to work with linear DNA, you would need a single site where all three of them could cut, because that means like basically you could cut it there and it would like be like isomorphic to like a linear piece of DNA. But yeah, so yeah, just general takeaways for part C questions. Make sure you read the questions thoroughly and then don't get scared away if there's too much text. But then again, also make sure to budget your time well. So if you think you are like getting too bogged down in a problem, it might be good to either return to it later or just like give a best guess and move on, because there are a lot of points that can be had elsewhere. Especially with math problems, make sure you think about like the extremes. So like intercepts, like when you like take a variable to infinity, how to sort of, yeah, like how those like relate, or even if they make sense in the context. And then also just taking logic puzzles piece by piece, and then building towards a larger explanation. Those are all good ways to do part C problems. But yeah, I will now hand it over to Kevin and Jason, who will talk about grading.
Thank you so much. Just before we move on to Kevin and Jason, I will introduce them really quickly, which I have not yet. So Derek, let me just share my screen real quick. So for the rest of the webinar, which is around 10 minutes, Jason and Kevin will be going over the scoring section of the Part C exam. And Jason was a 2018 and 2019 USABO national finalist, as well as an IBO silver medalist. And he has a degree in molecular biology and statistics. We will also be hearing from Kevin, who is currently studying evolutionary genetics at University of Washington-Madison. So he was a 3-time USABO finalist. So let's just welcome our two speakers who will be talking about the scoring section of Part C.
Okay, awesome. Thanks, Leon. So I'm going to share my screen. So I'll go over the scoring for the first question that Derek went over, and then Kevin will talk about the second one. But kind of generally for scoring, once you upload your PDF to the AOPS system, each one of your questions will be graded by 2 separate people. And then and we make sure that all those scores agree. So if there's a particular question where one person gave you 1.5 points and someone else gave you 2 points, those 2 people will meet and kind of discuss and figure out what the exact number of points to give is. And all of our graders are either alums of the USABO, so a lot of them are past finalists, or they're people with strong science backgrounds, so they all really understand the questions and would be able to fairly give out partial credit. So definitely don't worry about that. So for this question, the first one that Derek went over, this is pretty straightforward. So usually on the graders end, we would have a very detailed grading rubric, and oftentimes we're looking for specific points that you would be talking about. So for this question, so the first part is pretty straightforward. So if you indicate curve C, you would receive 2 points for that. And then for the rationale, on the answer key here, it says that we're giving out 5 points for the rationale. So I didn't grade this question, so I'm not sure what the exact rubric is, but I'm guessing that since the explanation depends on like justifying both the x-intercept and the y-intercept, the points would probably be assigned something like 2 points for correctly discussing the x-intercept and 3 points for discussing the y-intercept. So that's just some arbitrary point values I assigned there. And then, so I guess just some general tips for part C, like obviously try to write legibly. It's really useful to box answers, especially if you have a point you're trying to get across, like make sure you put in a box. That makes it easier for the graders. And also like it's in your best interest to kind of like write as much as you can, because maybe one of the things that you write is what we're looking for in the rubric. But that being said, don't just like write random stuff, because oftentimes if like you're writing a lot of things that are incorrect, that can also count against you. And like don't worry about writing complete sentences. You can use bullet points. You can just as long as you're getting the idea across. Yeah. So Kevin will talk about the second question now.
Wow. Okay. So, hello everyone. I'm Kevin. I had a distinct pleasure of presenting a question where we have no key and we have no history of how it was graded. But first I will say is that I know a lot of people are really worried about, oh well, I do a test and I write the answer is I write the transcontinental railroad and the answer is the Union Pacific Railroads. Well, I get partial credit. And again, like Jason said, we try our best to be as generous as possible. I will really say that like myself and other graders, despite the fact that we have like hundreds of students to grade, like we will go above and beyond. I remember there was this one student who submitted chicken scratch and I could not read it, but like the person I was grading with sent me a image where they rewrote in all their answers and reconstructed it. So that I'm not going to encourage you to do that, because some of them we just couldn't do anything. But if you try your best, we know you put a lot of effort and we do really appreciate that. For a question like this, I know everyone is pretty concerned about how partial credit will be given. I think for question A here, it'll be pretty straightforward, right? One part, if you say, no, it will not be cut, that's 1 point. And the second point will probably be why. Depending on the year, again, we don't have a key to base this off of. I think you would have to mention the effect of the fact that nutrition enzymes, because they function in bacteria, they specifically recognize methylation tags as a defense against self-damage to DNA by nutrition enzymes, right? Derek had mentioned earlier that they do serve as the immune system for the bacteria, and because viruses lack DNA methylation machinery, bacteria use that machinery to protect their own DNA. So it's a form of self-recognition. And I do think if the key says something to that effect, you would have to mention something about bacteria methylated DNA or nutrition enzymes cannot cut methylated DNA. There are some exceptions, but all three of these are traditional bacterially derived nutrition enzymes, which you can sort of tell from their names, right? And again, like we talked about, the names are derived again from the scientific names of the bacteria. Depending on the strictness, right? Maybe you had to mention the fact that it's usually the first letter of the genus name, the first 2 to 3 letters of their species name, and then a identifier for strain at times. For example, if you guys know the really famous enzyme ECO-R1, that's E. coli strain type R, and that was the first enzyme isolated from it. So the number would have to tell you where you got it. And the biological relevance, of course, like we mentioned, is that they do serve to cut viral DNA. For a question like B, it's only worth 1 point. It may be split up into 2 half points. It may be graded all or nothing. that is generally left up, if it's not mentioned in the key, up to the discretion of the graders. Like I said, we try to be generous, but we also have to be consistent. There's no way we can give a quarter of a point, because at that point it starts becoming too much of a hassle. So, try your best to answer questions correctly and precisely, because if you say a bunch of stuff and some of it is wrong, we as graders will know what you're trying to do. We're not dumb. We won't give you points for that. I don't understand why there's this belief that somehow the test graders are not people. There's some unthinking robots that like grade them in a certain way, and you can trick it. We're not dumb. So something to keep in mind. And for something like expression map, like, and this is a 7-point question, it's obviously very bulky. Well, the easiest way to grade this is that there are 7 revision sites. And if you had put about 7, depending on if you did it linearly or circular, and if you put them in the right place or not. And I think that's pretty straightforward. Again, I know this question, there's some confusion about whether or not it's a circular chromosome or a linear chromosome. It actually, in this specific case, does work out both ways. We don't know how it was graded at the time in 2018. And look, if we see a bunch where we see the students are saying an answer consistently, we will go back and look at the question. It's not like we're thinking, no, the key's absolute. We're very generous. I did a question where the key was, in my opinion, incorrect. I discussed it with my co-grader and we changed the answer. We don't think the answer on the key was correct. So that's something we're very open to doing. When you scan your answers, make sure you upload them to the proper section. We oftentimes know the other questions, but sometimes we can't be sure which exact question you're looking at or trying to see. So it will be, make sure you upload it to the right section, because you upload something that doesn't make any sense. Sometimes we don't know what to do with it. Also, don't upload nothing. If you have a blank sheet of paper, don't upload it. You won't get points. It really just wastes our time when we're grading, because it takes a while to open your files and stuff. So if you have a blank paper, don't upload it. If you wrote something, don't say, oh, my answer's probably wrong, I won't upload it. No, if you actually have nothing, or don't upload your Chinese homework. Someone did that. It was just really frustrating because we couldn't tell if the handwriting was just really bad, but it turned out it was Chinese homework, so that was not particularly pleasant. I mean, it's funny in the moment, but it does waste our time, and so please don't do that. I would also say, if you're doing any questions that does have math or requires to draw a graph, right? Remember your basic science. Remember your sig figs, your units, your axes labels, your arrows. Arrows were appropriate. Those are all things we graded for. And if you didn't have one of them, it was a 1-point question. You got 0 points. We are fair graders, but we're also very strict. Actually, I think 2-point question. That's the point. But that's how the grading tends to go. Make sure your answers are precise and include all the elements that you would need to answer. An incomplete answer may get you partial credit, but it may not be particularly generous depending on the question and how the points are allotted for that specific question.
All right. Thank you so much to all the speakers. We will not have time to go over specific questions about content of the exam or anything, but I did want to end just by asking Ms. Ring. I know a lot of people were asking about the points. So part A and B are all or nothing, even if there's like multiple select. And part C is where the partial credit comes in. And I think a lot of people have questions about accessing the exam again. So Ms. Ring, could you go over like whether they will be getting information from their proctors, or like what steps they have to specifically do to access the exam?
They'll get specific information from their proctors. They're also going to get an email from me with some of the steps as well, as they did last year. To access it, remember, go to the Art of Problem Solving site. You can use that link that I had that ends in semifinal, and it takes you directly to where the semifinal exam will be. And you can also find the practice session that will be up there tomorrow on how to upload it. So for all of you that are worried that you're not registered, go online tomorrow and access that practice exam session, and you'll know you're fine for the exam. All right. You are registered. Don't worry about that part. All right. Your ID was sent over to Art of Problem Solving if you were invited. All right, as a semifinalist. So no worries there. And as Leanne said, the point distribution, it's 1 point all or nothing for the first part, and the second part, multiple answers. There's no partial points. It's 2 points or nothing. In the semifinal, excuse me, the part C, that is strictly whatever the point values are by each of the questions that are asked within that question itself. Right? Don't stress out so much about that. Relax and, take a good look at what you have studied all this time, and don't cram. All right, get a good night's sleep and rest, and we look forward to seeing you next Thursday.
Well, thank you so much. Thank you so much to all the speakers, and we all wish everyone the best of luck on the exam next week. And if you had any questions that you didn't get answered, or you would like to ask more questions in the coming days, feel free to contact any of the emails listed here on the screen. These are all of our speakers and Mrs. Frame's contact information. And thank you again for everyone's evening time here with us.